things that I want to talk about this morning in lecture, and then I have a couple of things to talk about in lab, and we'll essentially be finished. Yesterday we finished with about an hour or so to study at the end of the lab, so I would anticipate much the same today. You guys are actually a little tiny bit ahead of where my other lab was, so you might even finish with a little bit more time. So I think we're in pretty good shape. So just a couple of quick things, kind of housekeeping sort of things. I got problem sets graded yesterday, so you should see your Unit 4 problem set score in Canvas. There's not a score there. There was only one or two where, I don't know why this happens. I'm sure that it isn't intentional, where it looks like people upload it and then it's like like a blank question sheet. That's happened a couple of times this semester, and like I said, I know it's not intentional, but so I can't see your answers. So if I commented that I can't see your answers, that's very likely why. And then I think there was one person who uploaded question 5 or Unit 5's answers instead of Unit 4. So just a couple of little things. So if you don't have a grade for that, I very likely made a comment in Canvas. So take a look at that and send me the right file, or send me the complete answers, and I'll be happy to update that grade in Canvas. Additionally, I've put in to Canvas all of the assignments for the rest of the semester. I put in Lab Exam 5 and I put in Lecture Exam 5. And of course, your Unit 5 problem set is in there as well. So you should be able to play the what if game at this point, if you're so inclined to do that. Play the what if game and see where things are likely going to land. I don't mean to sound glib. One sec. I don't mean to sound glib, but at this point, the only thing that you can do is your very, very best, right? So just do your best, lay it all out on the table, study your butt off for the next couple of weeks, and then we'll see where things land, right? So I can appreciate the what if feature in Canvas, but at the same time, I don't know. I would say go study instead. That would be my advice, but I don't mean that to sound glib. It's just at this point you can only do your very best on everything that's left. Yeah.
I was gonna say, is Canvas pretty accurate? Because
should be.
Sometimes it's okay.
I mean, it doesn't mess up the math too much.
Okay.
Are the Unit 5 problem sets showing zeros right now, or are they showing like just no score? Okay, I'll fix that. I'll take those zeros out so they show no score, and then you should be able to go in. Can you go in and change the 0 to something else?
Yeah, you can.
Oh, okay, then I'm not going to mess with it. Yeah, go in and change it to a 25, since that's likely what you're going to get if you play with the what if, and then you should be able to play with Lab Exam 1 or Lab Exam 5 and Lecture Exam 5. We might have a quiz next week. If we do that, that would obviously be something that gets added to the grade book. I did go in and fill in zeros for missing quizzes this week as well. So the quizzes are set up to drop your lowest 2. So many scores didn't change when I did that. A couple actually went up when I did that. So just kind of keep that in mind as well. So everything should be pretty close to up to date. Over the break, is a great time to work on your semester project. It's also, a great time to study and stuff like that. So, it's a lot of time off. It can be used to your advantage or your disadvantage. So, try to use it to your advantage. All right. There we go. And also on Canvas the other day, I put up, I think I did it yesterday, I put up an announcement with a Unit 5 list of topics. So you guys can use that to study over the break as well. So you guys have that available to you. So lots of information in Canvas right now. Lots of sort of end of the semester housekeeping that's coming up. Any questions about anything? Any other questions? Canvas should be pretty accurate though. I mean, it's just doing what I would do, which is calculating percentages. So, I've got all the rules set up, like dropping your lowest quizzes and stuff. So it should work pretty darn well. I always pull it into Excel and I double check it at the end of the semester, but I don't think I've ever really found an error. Yeah.
I saw that I put in the wrong document.
You did.
Yeah.
Yeah, you're lucky I didn't just drop you from the class.
It's still gonna be marked as later, is it?
Just get it to me. Yeah. You turned it in on time. You just turned in the wrong thing. So
I saw it.
just send it to me email, or just upload it to Canvas or send it to me via email. All right. Anything else? All right. Anybody do anything fun for Thanksgiving? I mean, I just told you to study the whole regs. So that's really the only right answer at this point. Anybody? Anybody? No? Just hanging out?
Thanksgiving.
Huh? What's that?
Is it Thanksgiving on the 28th?
It's on Thursday, yeah.
Next week.
It's less than a week away.
Just the normal stuff.
Just stuff, yeah. That's normal stuff. All right. Cool beans. All right. Well, I hope that you guys do have a great Thanksgiving break, and try to balance that. Excuse me. Try to balance that time off with the work that needs to get done, and all should work out well. All right. All right. So I think we had left off talking about this on Wednesday. Talking a little bit about the duodenum and all of the stuff that's being secreted. Right? Remember when that chyme that is ejected from the stomach into the small intestine. Remember that it is kind of only partially digested. Right? We've started to break down carbohydrates. We've started to break down fats. We've started to break down proteins, but nothing is completed. So as that chyme travels through the duodenum and into the jejunum, what's going to happen is that we're going to start to completely break those things down, right? So one of the things that I wanted to do today is look at how they're absorbed in the ileum, because the absorption of these major groups of macromolecules differs pretty considerably from group to group. And the first one that I think I'd like to talk about are fats. Fats have a really interesting way in which they're digested. In order to break down fats at all, we sort of have to break them apart, right? Remember that your structure of your fats. Turn the lights on just a little bit here. This is buzzwords. Remember that a structure of a fat is such that you have a molecule of glycerol, okay, and you have those three fatty acid chains that are attached to it, right? And the structure of that fatty acid chain is that it's this long hydrocarbon that we have like this. Because that hydrocarbon and that glycerol molecule are very low in oxygen, this molecule is what we call nonpolar, and it tends not to dissolve well in water. We call that hydrophobic, right? We've talked about this for quite a while. Excuse me. So I think we all know what happens when we try to dissolve fat in an aqueous solution like our digestive, right? All you have to do is cook a fatty meal and put water in that pan after you're done with that. You watch those fat globules or those fat droplets float to the top, right? Many of you will be doing that next week after you cook your turkey or whatever else it is that you cook on Thanksgiving, right? So it's not really a surprise then that that's exactly what happens in our gut. Is the fat that we eat forms these fairly large globules. It's really hard for enzymes to get in, those lipases to get in and cleave off these individual fatty acids when these fats are all globbed up in these big droplets. So what we have to do is we have to break those droplets up somehow. We have to have to do what we call emulsify those droplets. You do this when you do dishes. What do you use to emulsify fat droplets when you do dishes?
Soap.
You use dish soap, right? You use detergent. What that does is it breaks those fat droplets up so that you can wash them off of your dish and wash them away and leave it nice and clean. I don't recommend drinking dish soap. I don't think that would actually end very well for you. But we do kind of make our own emulsifier in the form of bile. So bile is a substance that's produced by the liver. It's stored in the gallbladder. We'll take a closer look at the ducts by which it's delivered to the small intestine today. But it does come from the liver. It's ejected primarily when we eat a fatty meal. And what it does is it causes these big droplets of fats to become emulsified, right? It breaks them up into smaller bits. And what that does is it allows enzymes like lipases to get in there and actually break them into smaller things. So once our fats have been emulsified, then lipase can come in and it can start to cleave off, right? It can start to cleave off these fatty acids. So we have lipase coming in and doing that. But we have to be able to emulsify it first. So what we're left with then, upon the breakdown of these molecules, is we're left with things called monoglycerides. So we only, notice how I only cleaved off two, right? So we have glycerol. This is called a monoglyceride. Mono meaning one, because we have one fatty acid. Plus, those fatty acids that we cleaved off, we call those free fatty acids. Okay, so this is essentially what lipases work to do, is they work to cleave off free fatty acids and then these monoglycerides. So we essentially are going to break them down. Now, I had mentioned very briefly on Wednesday that despite the fact that we're using these enzymes, these lipases, to break down fats, what's happening is that they're still pretty big molecules. Remember, some of these fatty acids can be up to 20 or 22 carbons long. So we're talking about big, long molecules here. So even though we're breaking this guy down into smaller bits, we're still talking about substantially sized molecules here. So they are absorbed in kind of an interesting way. We are going to emulsify them in bile salts. And when they become surrounded by bile salts, right, when we surround these guys by bile salts to emulsify them, once they've been digested, these are our bile salts here that I'm kind of putting around, right? We're surrounding them like this. They get kind of globbed together back again into these structures called micelles. Okay? Sometimes they're called bile micelles. Okay? Sometimes they're called bile micelles, but these micelles or bile micelles are how we sort of emulsify these guys. And what that does is these smaller globs of free fatty acids and monoglycerides can then get absorbed into the gut epithelium. So right here, we're looking at a gut epithelial cell. You can see the little microvilli on the brush border right here. And we see here that we're absorbing those free fatty acids and the monoglycerides into the cell. Okay? So just really simple absorption, but we have to kind of package them into these things called bile micelles in order to be able to do that. What we do next is a little bit counterintuitive. Once we get the free fatty acids and the monoglycerides into the intestinal epithelial cell, what we do is we actually will reverse this reaction and we'll resynthesize the fat. Okay? We'll resynthesize the fat. So once we're into the epithelium, what we do is we reverse the reaction and we actually will resynthesize the triglyceride. And this always feels a little bit backwards, right? I guess they're just, evolution hasn't given us a way to get the entire triglyceride into the cell. So we have to sort of break it apart, pull it into the epithelial cell, and then we rebuild it once it's in the intestinal epithelial cell. So we can see that right here. Once those bile me cells have been moved into the cell, what we do is we resynthesize them into triglycerides. Okay? And then what we do is we package them with proteins. Okay? We package them with proteins, and those are structures that we call chylomicrons. All right, so we package them with proteins. Let's go the other direction here. Get rid of our lipase. All right, so we've rebuilt this guy in the intestinal epithelial cell. Now we package them with proteins. Right? Package them with these long amino acid chains, these proteins, and we get things that are called chylomicrons.
Bless you.
Gesundheit. Chylomicrons are a combination of the proteins that we're looking at, and then we have those fatty acids inside of there, right? So there's a triglyceride, right? Here's a triglyceride right here, right? Something along those lines, right? So these chylomicrons are kind of a combination of these proteins and then these fatty acids that are all sort of clustered together, right? You probably know chylomicrons by a different name, because I bet you've heard of them. How many of you guys have heard of lipoproteins? A lipoprotein is a combination of a lipid and a protein, right? Often it's going to be cholesterol packaged in with these guys as well. We're kind of ignoring that right now, but there often is cholesterol packaged in with these. So one of the things that we do is we look at the ratio of protein to fat in those, and we calculate the density of the lipoprotein. We generally get two different kinds. We get high density lipoproteins, and we get low density lipoproteins. Your HDLs and your LDLs, right? The ratio of protein to fat tells us about the density of things. Which ones are the good ones? HDLs, the high density lipoproteins, right? So you're looking at a higher ratio of protein to fat when you look at those sorts of things. Fats are not terribly dense, but proteins can definitely be that way, right? So your HDLs are going to be the ones that you want. You want to avoid the LDLs, right? Those LDLs often come because we have a lot of unsaturated fats that are reducing the density of the packing, and that can be really, really problematic. Okay. So you guys have heard of these, you just haven't heard the name chylomicron before, or at least I'd be willing to bet that you haven't, right? So these chylomicrons are these big vesicles, essentially, of proteins and fats, right? They're big. Not only are the fats big, but now we're bundling a whole bunch of them together with all of these proteins, right? So in order to absorb those into our circulatory system, we've got to have a way to do that because our capillaries are simply too small, right? We're talking about big things here that aren't going to go into your capillaries well. So what we actually end up doing is we actually end up absorbing those chylomicrons into our lymphatic system. And remember, we looked on Wednesday at the structure of our small intestine, and we saw that all of the villi have that lymphatic vessel going into it called a lacteal. Right? So that lacteal is necessary because we absorb fats into our lymphatic system. Those are really big vessels. They're really permeable, I guess you could say. Just about anything fits inside of them. So what happens is that we can absorb just about anything into those. And then, of course, the lymphatic vessels will dump into much larger blood vessels closer to the heart, so we can kind of avoid the capillaries as much as we possibly can. So fats have a really, well, quite frankly, a bass-ackwards way into the circulatory system, right? We have to break them down to get into the gut epithelium, and then once we're in the gut epithelium, we rebuild them, and then we have to absorb them into the lymphatic system in order to dump them into the circulatory system. So, man, fats have a really long and circuitous route into use, essentially. It's very convoluted, yeah. There's a lot of steps to it, and they don't always make sense, but you know, that's the way evolution works. It doesn't always make sense, right? All right. So, because bile is so important for the breakdown of fat, if you have issues with your liver or your gallbladder, especially if you've had your gallbladder removed, your gallbladder is a giant storage organ for bile. If you've had your gallbladder removed, it's usually advisable to avoid fatty meals when that happens. Because you're not able to break down fats the way that you should be able to. People who have had their gallbladder removed, if they eat fatty meals, they often will complain about a lot of indigestion, upset stomach. Sometimes it can lead to diarrhea and all sorts of gut health problems. It's not a very pleasant experience. But you've got to have that bile in enough of a concentration to be able to emulsify the fat that you eat. Back in most of you are going to be way too young to remember this, because this happened in the 1900s. But back in the 1990s, towards the end of the century, they had developed this type of fat that they were able to fry potato chips in and stuff like that. It was marketed as a lean, and I guess the idea was that you could make potato chips in this, and you wouldn't lose any of the flavor. But for some reason, we weren't able to actually absorb the fat. We weren't able to break it down. We weren't able to absorb the fat. So the idea was that you would get the chips with all of the flavor, but you wouldn't absorb the fat, right? So, I mean, that seems like a win-win, right? You get to eat all the potato chips you want, and you don't actually absorb any of the fat. What could possibly go wrong? Well, that fat has to go somewhere, and it gets pretty greasy and loose towards the end of the digestive tract if it doesn't get absorbed. So people who were eating large amounts of these chips, and people were eating large amounts of these chips, they would go on the shelf in the grocery store and sell out within an hour, right? It was crazy. I remember it. My mom was all about them. And I tasted them. They actually tasted pretty good. I was in high school when this happened. So it was the mid-90s. Anyway, again, people were eating a lot of these chips, and that fat had to go somewhere. So they actually went so far as having to put a warning on the bag, and this was before they took it off the market. They put a warning on the bag that said, "Warning, may cause anal leakage," because that's exactly what it was doing. Is people's anuses were leaking like grease, basically, and not good. I even mentioned to my wife last night, I said, I get to talk about a lean tomorrow. And she says, oh yeah, the explosive diarrhea and greasy poop? And I said, yeah, because if you grew up in the 90s, you remember this. And it was astounding. As a high schooler who had the sense of humor of a 12-year-old, I thought it was hilarious. As a man in his mid-40s who still has the sense of humor of a 12-year-old, I still think it's hilarious. But yeah, so that's a fun fact. Kind of an interplay between marketing and digestive physiology, right? It seemed like a great idea at the time, but apparently nobody bothered to consult a physiologist in the production of these chips, because it did not end well for people who ate a lot of them. So they took them off the market. You can't buy them anymore, but they lasted a few short, glorious months. So anyway, like I said, it's the junction between digestive physiology and marketing. All right, so any questions about fats? Yeah.
You said this was happening in the helium?
Yes. All right. Let's talk a little bit about protein digestion. So remember that time I told you guys that you don't have to memorize all 20 amino acids? Yeah, you don't have to. And so we're not going to go through the absorption of all 20 amino acids. But someday you might have to do that, because there are 20 different amino acids. That means there's 20 different ways that they get absorbed. We're going to simplify it and talk about some generalities. So you have your polypeptides, okay? We know that in the stomach we're releasing pepsin. In the pancreas we're releasing a whole bunch of other enzymes that you do not need to learn the names of in order to break down those peptides. They are either broken down into free amino acids or shorter chains of amino acids that we call oligopeptides. And then eventually, most of them are broken down into amino acids. We do have the ability to absorb what we call dipeptides or even tripeptides, where you have just like 2 amino acids or 3 amino acids linked together. But the majority of these eventually are going to be broken down into amino acids, right? So the absorption of those guys is fairly straightforward. What we do is along with sodium, we just simply absorb those amino acids into our capillaries and send them along their merry way, all right? The same thing is true with dipeptides and tripeptides. There's not a whole lot to say about it, except that the absorption of those is also sodium dependent. You'll notice that along with the amino acids, sodium is also being absorbed. Along with those slightly longer chains of amino acids, sodium is also being absorbed. So if it wasn't for sodium, you wouldn't necessarily be able to absorb those guys. Incidentally, that's sort of another role that isotonic sodium chloride plays, that the duodenum secretes, is we are putting some sodium into the gut so that we can absorb these sorts of things a little bit later on. You'll notice the slide here says that the amino acids are being absorbed to the portal venous system. What's that? What's the portal venous system? The hepatic portal system, yeah, right? So we're actually going to have to take a little bit of a look later today at the hepatic portal system again, because it hasn't gone anywhere. It's still there, and we have to deal with it. So we're going to take a look at that a little bit later on today. But we are absorbing these into that hepatic portal system, and we'll take a little bit of a closer look at that today. All right? So fats are pretty backwards and convoluted in terms of their absorption. Amino acids are pretty straightforward. We just absorb them. It's just that we do need sodium to do that, right? They're a sodium-dependent absorption. And that's really about it. I mean, there's not a whole lot left to say about them. I do like that this slide also sort of guides you through the process of protein digestion. It explains what's happening in the stomach, what's being secreted from the pancreas, and then so on and so forth. So that way, so you can kind of guide through this as well. But we do get the absorption of these guys. So it is sodium dependent for the most part. Like I said, there's a lot more to it. All 20 amino acids have their own specific mechanism, but most of them are sodium dependent. All right. Let's talk for a couple of minutes about sugar. Sugar is absorbed into the gut epithelium as well. I think we've mentioned a couple of times that our primary monosaccharides that we end up with after we break down our polysaccharides and our disaccharides are glucose, galactose, and fructose. Just really quickly here. So we have our monosaccharides, glucose, galactose, and fructose. Glucose and galactose are both 6-carbon sugars. They both have the chemical formula C6H12O6. It just has the differences the way that certain things are turned on the molecule. Fructose is a 5-carbon sugar, so it has that chemical formula C5H10O5. But these are the primary monosaccharides that we end up with after we break down the things that we sort of normally eat. In order to absorb these guys into the blood, we need a protein called a glucose transporter. Okay? They're called glucose transporters, but they can transport more than glucose. They can transport just about any monosaccharide. Okay? So we have these proteins called glucose transporters. Because biochemists don't like to write names like glucose transporter out over and over and over again. They like to abbreviate things. So glucose transporter is usually abbreviated using the GLU from glucose and the T from transporter, and we get the name GLUT. So these are GLUT proteins that we look at. They're glucose transport proteins. And there's a whole family of GLUT proteins. Even though they might name them somewhat creatively, they don't diversify them creatively. They just go GLUT1, GLUT2, GLUT3, GLUT4, GLUT5. Probably in the order that they were discovered, or maybe the order they like them best. I don't really know. Don't ask me why my kids are numbered 1 and 2. It's either the order they were born in or the order I like them in best. I won't tell them, don't worry. It's a secret, it's safe with me. Nevertheless, though, they're not numbered very creatively. But they are found in different tissues, right? So in some tissues we find certain GLUT transporters, in other tissues we find other GLUT transporters, right? In the gut, the two that we're primarily concerned with are GLUT2 and GLUT5, all right? Let's look really quickly at fructose. It's a relatively simple mechanism. We notice that fructose, these little 5-carbon sugars that we have, are moved into the intestinal epithelium here by a GLUT5 protein. Okay, again, it's just a glucose transporter. Whoop, right across the membrane, there it goes. All right, it travels through the interstitial space of the, or not the interstitial, the intracellular space of the epithelial cell, and then it's transported across that basal membrane by a GLUT2 protein, and it goes, just diffuses into the blood, right? It doesn't get a whole lot more simple than that. You've got a couple of proteins moving these guys across, and life is very, very simple. Glucose and galactose are also fairly simple, but they involve a sodium-dependent mechanism. So they're kind of like amino acids. They involve a sodium-dependent mechanism. You'll notice that glucose and galactose are moved into the epithelium with a protein that requires sodium to work. All right, and then it's moved across the basal membrane that we see right here with a GLUT2 protein, just the same as fructose is, and then it diffuses into the blood. That protein that moves glucose and galactose into the cell, you'll notice on our little key right here, this little guy, this little blue circle is keyed out as something called SGLT. Okay, SGLT stands for sodium glucose transporter. You don't necessarily need to remember that, but that's what it stands for, right? It stands for sodium glucose transporter, SGLT. Okay. So we have sodium glucose transporter that move glucose and galactose across the cell membrane. Okay. And then they're transported. These are small molecules. They get transported into the capillaries, and they go into that hepatic portal system the same way that amino acids do. All right. All right. Any questions about proteins or carbohydrates and the way that they're absorbed? Yeah.
So GLUT5 moves it past the first part, and GLUT2 moves it past the basal membrane.
Yes.
Okay.
So on the luminal side of this guy, right, close to the lumen here, you find that fructose is moved across by GLUT5,
Okay.
and then on the basal side of these cells, we have a protein called GLUT2 that moves them into the blood.
Okay.
Are the 2 and 5 just there to distinguish the two different sugar molecules?
No, I don't think it is. I think it's there and it happens to move fructose apart, but I don't think it can't move glucose and galactose across. It's not able to do that just based on the structure of the molecule. So we had to have a different way for glucose and galactose to get into the cell, and apparently we evolved this SGLT protein.
So that protein is just in the epithelium, or is going together with the protein?
Do what now? I'm sorry?
The SGLT
Uh-huh.
stays there or comes together?
No, it's embedded on the membrane. Remember, all of our cell membranes have proteins embedded on them, right? So there's some of the proteins that are embedded are meant to transport things across. So this protein, this SGLT protein, is embedded on the luminal epithelium that we see here. Yeah.
So are you saying that like these proteins are kind of just like channels instead of like they're not actually doing the transport, they're just opening?
I'd have to look. I think like SGLT is a transport protein. What it does is it actually has a shape change. Like if this is the lumen of my small intestine, it will be like this. Sodium and glucose, for example, will go in, and then it actually changes shape and does this to let them out on the other side. So it actually is it's not just an open channel that allows them to go across. It actually does have a change in shape when those things bind to it. But it won't change shape unless sodium happens to be there as well, right? So if only glucose went in, it would just sit there like this until a sodium goes in, then it changes shape and lets them out on the other side. I'd have to look it up. I'm honestly not entirely sure what the mechanism of GLUT5 is. I don't know if it's a channel or if it's an actual shape changing protein. I don't really know. I'd have to look that one up. Kind of interesting, right? Sodium becomes important yet again, right? Sodium is important for so much that we do. It's necessary for action potentials. We need it for the absorption of amino acids and sugars. We need sodium for reabsorption in the kidney. We looked at that in our last unit. Sodium is important for so much that we do. It's really remarkable, actually. All right. Anything else? I wish it was kind of a cooler story. I wish I could wow you. We've spent a lot of time getting to this point. I wish it was kind of a cooler story, and I'm just like, yeah, they're absorbed. Just real quick, I want to kind of mention a couple of things. Just to reiterate the fact that the movement of food in the small intestine is just so incredibly tightly regulated. As soon as we start to eject food or chyme into the small intestine, we start to regulate it really quickly. I just put a couple of things here to sort of give you guys an idea here. So secretin is a hormone that inhibits stomach acid secretion and really slows gastric emptying. And then it stimulates those duodenal glands to secrete bicarbonate. So as soon as the stomach gets full, we start to really regulate how quickly we empty the stomach, and secretin is one of these hormones that's responsible for that. Another one that we see that's released is a chemical called cholecystokinin. Cholecystokinin is secreted when meals contain fat. It stimulates gallbladder contraction, but it also slows the emptying of the stomach. And this is really important because that breakdown of fats, as we saw this morning, is a really, really slow process. Fatty meals take us a long time to digest. So we have to really slow things down, and we have to let the gut do its thing over a longer period of time. Two additional ones that we see are gastric inhibitory peptide and glucagon-like peptide. Those are released when we have meals that are rich in carbohydrates. They promote the release of insulin, and they also slow gastric emptying. So those are what we see with carbohydrate-rich meals. I'm not entirely sure, to be quite honest with you, how our gut senses what our meals are made of, right? But they do seem to be able to sense if we have a meal that's rich in fat, and we can promote gallbladder contraction if that's the case. And then we also can sense if we have a meal that's rich in carbohydrates. But I'm not entirely sure how our digestive system senses that and sort of moves on. That would be sort of an interesting thing to look at, Okay. All right. So we move through the ileum, we absorb our fats, we absorb our carbohydrates, we absorb our amino acids, and then we eventually get into the large intestine, right? The large intestine is kind of a weird organ. It's obviously not anywhere near as long as the small intestine is. And our view of it has really changed over probably the last 20 to 25 years. If you had taken this class 25 years ago, I would have told you that the large intestine does three things, right? It's responsible for the absorption of vitamins. We know that that's true. We haven't really talked a whole lot about vitamin absorption yet, but that's the large intestine is where most of it happens. You can also make vitamins in your large intestine. One thing that we do know about the gut is that it has a really big population of bacteria. We're talking hundreds of species of bacteria living in your gut. You're made up of about 75 trillion cells. Your gut houses about 10 times as many bacterial cells, about 750 trillion bacterial cells living in your gut. So it's a huge, huge number. You have about as many more living on your skin. So it's a really remarkable ecosystem that we sort of house. And that's a little bit beyond the scope of this class, but it's kind of an interesting one. And the other thing that we would have told you that the large intestine does is it absorbs water. Okay? Remember, we've been doing a lot of secreting at this point. Saliva, stomach acid, bicarbonate, a ton of mucus has been secreted. We've got to absorb all of that water back into our circulatory system so that we don't become dehydrated, right? And the large intestine is where a lot of water absorption occurs, okay? Kind of a side note to that, right? If digestion moves through your large intestine too fast, you don't absorb very much water, and you can get diarrhea as a result of that, watery stool. If you aren't moving things fast enough through your gut, and they take a really long time to move through, you can actually absorb too much water, and your feces become very hard and compacted and difficult to pass. We call that constipation, right? So just like Goldilocks and the Three Bears, you have to move things through your gut at just the right speed so that you absorb just the right amount of water so that it's easy to poop. that would be a different children's book to write, so we're not going to write that one. But those have kind of traditionally been the three things that we viewed the large intestine as doing, right? Absorbing water, absorbing vitamins, and then we know that those bacteria produce vitamins just a little bit, all right? But one of the sort of lingering questions in digestive physiology for a really long time has been, why are there so many bacteria in our gut? What else are they doing? Because we've seen sort of, I don't want to say accidental, right? But we've seen sort of anecdotal things about what happens when those gut bacteria are depleted and things like that. One of the things that we have learned from other animals is that they, especially herbivores, ones that eat a lot of plant matter, is that they have to be able to get as many nutrients as possible out of that plant matter. And one of the things that we know about them is that they have guts that enable them to do that. And they do a huge amount of fermentation. Right? Fermentation takes place somewhere in the digestive tract in herbivores. And what it does is it allows them to convert these carbohydrates into lactate and short-chain fatty acids that can be absorbed by diffusion. And if we look at the digestive tract of certain animals, we see that that's definitely the case. This rabbit up here is a really, really good example of this. The rabbits, as you guys probably are aware, are herbivores. They have a fairly normal-sized stomach, but if we follow their gut all the way through, we see that they have a really, really big cecum, okay? Like a massive cecum, like 3 or 4 times the size of their stomach. If we look at mice and rats or horses, we see that that's also the case with them as well. A massive cecum compared to their stomach. We look at other herbivores, like this deer that we see right here, and this deer has a fairly normal-sized cecum, probably bigger than ours, but not really remarkable. But it's got a massive stomach. In fact, that stomach has 4 distinct chambers to it. So we've known for a very long time that these animals, whether they have a big cecum or they have a big stomach, are using that space to house bacteria that will ferment the plant matter that they eat and extract those short-chain fatty acids and things like that from the food. Which is something that for a long time we thought we didn't really have the ability to do because we don't have a big cecum, we don't have a terribly large stomach, right? So what we've learned though over the last 20 or 25 years is that we actually probably are doing a lot of that in our large intestine, and it's probably a lot more significant than we thought 20 or 25 years ago. It's this fermentation that's happening in our gut, right? So we probably are actually able to break down a lot more plant matter than we originally thought using the bacteria in our gut to extract those short chain fatty acids and lactate and things like that. So it's kind of an interesting thing that we've learned. This is why, from a dietary point of view, we might have failed with the Olean chips, but we've done a good job of marketing prebiotics and probiotics, right? Prebiotics would create an environment in your gut that would encourage bacteria to grow, and probiotics would encourage the growth of that bacteria, right? And our diets should be rich with those things so that we can maintain a healthy gut flora and hopefully be able to make the most use of this. We've also been able to link a healthy gut bacterial population to mental health and all sorts of other things, right? That's called the gut-brain axis. And there's really, really strong and very convincing links to mental health and things like that. So there's all sorts of things that we're still continuing to learn about the bacteria that live in our gut, but they appear to be far more important than we thought about 20 or 25 years ago, right? 20 years ago, we were just like, yeah, you've got them. They appear to do something, and we don't really know what. But we're learning a lot about these guys. So kind of an interesting story that's still developing, and it's definitely something that you probably we'll learn more about in micro and stuff like that. So, just real quick, back to our other animal friends here. You know, cows are a great example of what we call a ruminant, where they have these really big four-chambered stomachs. The breakdown of that material takes forever and ever and ever. So if you hang out with cows long enough, you usually will see that they appear to be chewing something just about all the time. What they'll do is they'll actually bring this food into their gut, all of this plant material, you know, and they'll let the bacteria break it down. But there's a lot of mechanical digestion involved. So what they'll do is they'll actually regurgitate little bits of it, chew it again, and then swallow it, and regurgitate and chew and swallow, right? They call it chewing their cud. But if you ever see a cow, it looks like they're always chewing something. It's because they are, right? They're just regurgitating and chewing, regurgitating and chewing, and continuing to break it down. As that material changes density, what it does is it falls into different parts of the stomach where further chemical breakdown can happen. But it's kind of an interesting one. And they do a very good job. It just takes a really long time for them to be able to break those things down. Obviously, animals with a big cecum don't do that regurgitation. So you don't see horses chewing their cud in the same way because they can't get it from the cecum all the way up. And they aren't as good at breaking it down either as a result of that. So they have to eat higher quality plant materials. So if you've ever fed cows and horses, you know you have to feed them slightly different food because horses need a slightly higher quality plant material than cows do. So it's kind of an interesting one. Rabbits are also interesting because they do this, but they also do something else, right? They have evolved a behavior in which they actually will send their food through their gut 2 complete times. So rabbits are what we call coprophagic. Does anybody know what it means to be coprophagic? You know what phage means? Or phagic? Like a macrophage? It means to eat or consume. You know what copro means? You know what a coprolite is? Any dinosaur geeks? No? A coprolite is a fossilized dinosaur poo, okay? So coprophagic means feces eating, essentially. Rabbits produce 2 kinds of feces. They produce a very soft feces that they eat directly from their anus. And then what they do is they send it through their gut a second time, extracting more nutrients from it, and then they produce a second hard pellet that is what you see when you have rabbits, is those hard pellets that come out. But that's the second time around. That's recycled poo. So, happy Easter, everyone. That's the Easter bunny. Those are Easter eggs, by the way. Those are Easter eggs. At least it makes you look real hard at your Easter eggs, doesn't it? That was the day Dr. Russell ruined Easter for me. But they sure are cute, man. Rabbits are stinky cute. All right. Huh? Don't trust the chocolate eggs. All right, so by the end of this, you have feces that are moved into your rectum. Feces just by the very nature of them moving through the large intestine, are about 50% bacteria by weight. So there's a lot of bacteria in our feces. Fortunately, it replicates really fast in our gut, so we're all good there. But you have undigested plant material, undigested food. You have obviously the breakdown of red blood cells pigments your waste and stuff like that as well. So you have all of this waste being moved into your rectum. Now, the rectum is usually closed off by 2 sphincter muscles. In lab, you guys looked at the internal and the external anal sphincter muscles. These are different muscles, not only in name, but also in structure. Your internal anal sphincter muscle is really just a continuation of that circular layer of muscle in your gut, so it's smooth muscle. The external anal sphincter is skeletal muscle, okay? But both of these muscles stay contracted when the rectum is empty. They close off the entrance to the anus from the external environment, right? So we have that. But what happens is as feces are moved into the rectum, it causes that rectum to distend or stretch. And we trigger stretch receptors when that happens, and those stretch receptors send a message to the spinal cord. The spinal cord sends a message back saying, relax the sphincters, okay? Relax the sphincters, okay? And the sphincter muscles relax, and the rectum is emptied, okay? So the defecation reflex is nothing but a spinal reflex, all right? Very, very simple. You stretch the rectum, the sphincters relax, and it empties, okay? But you know, and I know, that we live in a society where not every time is a good time to empty your rectum, okay? In fact, right now is not a great time to empty your rectum, okay? So you need to learn to hold it, right? So as little kids, we go through potty training, right? And the idea with potty training is to recognize what it feels like to have a full rectum and to be able to essentially override that spinal reflex, right? We can't do anything about the internal anal sphincter. The internal anal sphincter is smooth muscle. You have no conscious control over it. But you can, with just a little bit of practice, you can learn to control the external anal sphincter because it's made of skeletal muscle. Leave it contracted until you have time to make it to the bathroom, okay? So as potty training, in potty training, we learn to essentially control that external anal sphincter, and that allows us to make it to the bathroom without incident. All right. We didn't quite get to it with the kidneys, but urination is the exact same way. You have two sphincter muscles around your urethra. One of them is internal. It's the internal urethral sphincter. It's made of smooth muscle. The other is the external urethral sphincter. It's made of skeletal muscle. And the idea is exactly the same. When the bladder fills with urine, it stretches, triggers a stretch receptor. You have a spinal reflex that causes you to relax those sphincter muscles, and you would empty your bladder. But again, through potty training, you learn to control the external sphincter, which is made of skeletal muscle, and you can actually hold the urine in the bladder until you're in an appropriate place to empty it. So the idea is exactly the same with urination as it is in defecation. Okay? So easy we can even teach dogs to do it. All right. Any questions? And we made it through digestion. We just have a little bit of anatomy left to go through in lab, and then we're good to go. All right. And we're on time, 9 o'clock. So I'll see you guys in the lab.